So now that we understand the genetic code, how these 3 bases code for a specific amino acid, now we're ready to understand these processes, transcription and translation. So in this screencast, we're going to focus on transcription, which is this act of using the information in DNA to generate this structure called mRNA. And again, think of this like we're making a photocopy of a subset of the information of DNA. In other words, DNA is the cookbook, mRNA is the recipe, and we're making a photocopy of that. So given that, where is that going to occur? Did you say the nucleus? Because it's in the nucleus. Because remember, the DNA can't leave the nucleus. In other words, the cookbook is not allowed out of the library. So how does this go down? I do think it is worth hitting pause and watching this animation. It's linked from the slideshow. Just watch until about 1:35 seconds, and this video will just kind of give you a nice overview of everything that I'm about to discuss. It's kind of helpful to see it in 3D. So I'll leave that to you. But we're going to do player's process here, and again, having seen that animation would help make sense of what we got going on. So the first player is something called ribonucleic acid, or RNA. We've kind of briefly mentioned RNA here and there as an example of a nucleic acid, but we haven't really said much about it. So now we're going to dive in. So what is the importance of RNA, ribonucleic acid? Well, first of all, it allows the cell to code specific regions of DNA that are specific to the needs of the cell. In other words, DNA is the cookbook, messenger RNA is the single recipe for the pie. And I think in my analogy, I used cake. Whoops. Anyway, so, an interesting question is that every single cell in your body has the exact same DNA, minus mutations, but for the most part, the exact same DNA. So how is it that you have all these different cell types? Some of your cells are skin cells, some are nerve cells, some are muscle cells. How does that work? Well, the answer is the same DNA is in every cell, but we only bake certain recipes in each cell. And so the skin cell gets the skin recipes, the muscle cell gets the muscle recipes. And so by coding different RNA molecules, we can actually have each cell have its own unique identity. A lot more on that in the next chapter. Second, it's kind of a disposable copy of genetic information. The cell doesn't need to worry about damaging it. Since it doesn't last very long, it can be remade. And as a result, we can fiddle around with it through these processes, transcription, translation, without worrying about damaging like the core information. So, DNA is kind of like, it's like the original constitution that was written out by our founding fathers. You don't want to be messing around with that document too much. You want to keep it nice and safe, stored in the nucleus, and then we can make copies that we can mess around with and play around with. Okay, the structure of RNA. I do think it's worth copying this table down because it's pretty important. And I have pictures of all of this on the next slide. So first of all, DNA is extraordinarily stable. We have found DNA intact from like woolly mammoths that have been, frozen in ice in Siberia. Tens of thousands of years ago. They've even found little tiny snippets of DNA from dinosaurs. So it is extraordinarily stable. RNA, not so much, breaks down very quickly. The sugar group is different in these two. So in DNA, the sugar is what is called deoxyribose, hence deoxyribonucleic acid. RNA, it's a sugar group called ribose, which is ribonucleic acid. So here's two pictures that show the sugar groups, and you may recall that every nucleic acid, the building blocks of our nucleotides, and each nucleotide is a sugar, a phosphate group, and a base. So here's the sugar group, and deoxyribose has a hydrogen here on this particular carbon. Ribose has an OH group. So the name deoxyribo refers to the fact that we've kind of removed this OH group and replaced it with an H. It turns out that, well, actually you already saw that, well, no, you didn't, just kidding, okay. Anyway, we'll move on. So that OH group is what's different. So ribonucleic acid versus deoxyribo. The base pairs are a little bit different too. We've already seen that in DNA, the bases are A, T, C, and G. In RNA, it's A, U, C and G. So here for RNA, here's uracil, and you can see it looks a lot like thymine, but it's slightly different. And I don't know if there's a particular reason why this happened, or it's just kind of an accident of evolution, but for whatever reason, it's uracil instead of thymine here. So U pairs with A instead of T. Number of strands, DNA is double-stranded, RNA is single-stranded. And then the length, DNA is very long because it's the whole cookbook, whereas RNA is very short because it's a single recipe. And then finally, DNA lives in the nucleus, while RNA can kind of go back and forth between the nucleus and cytoplasm. Since it's much shorter and smaller, it can pass through those nuclear pores. Here's a lovely picture for you Game of Thrones people out there of DNA versus RNA. Okay, different types. There's actually different types of RNA. So messenger RNA is one that we've kind of alluded to already with my cake/pie analogy. And this is a disposable copy of genetic information. It relays the genetic code information from the nucleus to the ribosome, hence the name messenger. There's a couple others, though, that are important. Ribosomal RNA, or rRNA, this is a structural component of ribosomes. So ribosomes are made of both proteins and RNA, and so that ribosomal RNA is in there. We don't talk much about that, but it's an important structural piece. And then finally, there's this thing called tRNA, or transfer RNA. It looks kind of bizarre like this. This plays a huge role in translation, which we're going to talk about in another screencast. The second player after RNA is RNA polymerase. This is the enzyme responsible for synthesizing messenger RNA using DNA as a template. Now, if you look at this enzyme should remind you of another enzyme, both in looks and name, DNA polymerase. And essentially, it acts in a very similar fashion, only it makes mRNA instead of DNA. But you can see it similarly kind of splits the DNA apart and sort of builds the new chain of mRNA. The next player is the DNA itself. The DNA has the information, and there's some features that we got to talk about. So first, on the DNA, a lot of people think that DNA just codes for stuff and that's it, but there's actually tons of regulatory regions that don't actually code for anything, but rather just help the machinery of the cell make things out of that DNA. So one of these regulatory regions is something called a promoter. This is the region of DNA where transcription actually begins. And it contains something we call the TATA box, T-A-T-A box. And it's this region that's really rich in T and A nucleotides. This is the point where transcription begins and the two strands are sort of unwound for the first time. Now, think back to what we talked about in the last chapter about the T and A nucleotides versus C and G. How many hydrogen bonds hold T and A together versus C and G? Do you remember? T and A only had 2 hydrogen bonds, while C and G nucleotides had 3. That means that T and A are not held together quite as strongly as the C and G nucleotides. And so if we're trying to unwind the strand, this is a logical thing to do. Use a bunch of T and A's because they're a little bit weaker, and then we can pull those apart. And so here's just kind of a picture of the A to T hydrogen bonding and the C and G bonding. It also contains binding sites for what are called transcription factors. These are regulatory proteins that simply aid in the binding of RNA polymerase. In other words, this RNA polymerase enzyme needs these things in place so it can be positioned in exactly the right spot. Here's the process. So step 1, RNA polymerase, that enzyme, is going to bind to the promoter region. Again, it relies on those transcription factors to be in the right position so that it can be exactly where it needs to be. RNA polymerase then unwinds the strands, and then it adds RNA bases of the new strand in the 5' to 3' direction. So you'll note that RNA polymerase can both unwind the strand and initiate the production of the new strand. So in this sense, RNA polymerase is like a way better enzyme than DNA polymerase. You may remember DNA polymerase required primase to lay that little RNA primer down, and then it needed helicase to unwind the 2 strands. RNA polymerase does all that stuff just by itself. So it's going to unwind, and it's going to start, and it's going to build. And note that it's always adding to the 3' end of the new strand because that's where that hydroxyl group is for the dehydration synthesis reactions. Note also that if there's an A in the DNA, U binds to it. And if there's a T in the DNA, then A binds to it. So it just, anytime you're working with mRNA, it's a U, but if you're working with DNA, it's a T. So RNA polymerase is just chugging along here, you know, C, U, U, A, G, C. And then eventually it's going to reach a specific DNA sequence, at which point it's going to stop. RNA polymerase is going to detach. And then at that point, the mRNA is finished. So that's pretty much it. So it just attaches, unwinds the 2 strands, builds the next one, and then closes back up again. That's it. Note, by the way, that only one of the 2 DNA strands is transcribed. This strand is called the template, non-coding, or antisense strand. And so you can kind of see like this strand right here, this strand, I'm sorry, in yellow is what's actually transcribed. A, U, G, 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 G. And so this is the antisense strand because the ultimate mRNA is the same, essentially, other than T substituted for U's, as this original strand, which is the sense. And so, I don't know, the name makes sense, but it's kind of confusing. So just a minor point. All right, here's that same animation I recommended that you watch at the beginning. Here it is again, in case you ignored me. I still think it's worth doing as kind of a recap of what we just talked about. The final thing I want to talk about with regard to transcription is processing. So in eukaryotes, the mRNA is processed after transcription. Think of this like adding 3-hole punchers or stapling your pages of your copies together. So the first way it's processed is the mRNA ends are modified. This little 5' cap, it's basically a modified guanine nucleotide. It's added to the 5' end in the reverse orientation. So it's just this little extra chemical reaction that happens. The reason why is it helps the mRNA exit the nuclear pore and fit into the ribosome. So it's like, oh, the mRNA is done? Great, let's add that little 5' cap. There it is. Okay, that's it. This is what it looks like in a more biochemical sense, if you're curious. On the 3' end, this poly A tail is added. Just a whole bunch of A nucleotides are added. The reason why is because enzymes, remember, mRNA is not very stable in the cell. Enzymes are going to start to break this thing down from the 3' end almost immediately as soon as it's made. So by having this poly A tail, it means that these enzymes that are, eventually degrading this mRNA, they're only going to be attacking this poly A tail at first, right? Chomp, 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 chomp. And so then that preserves the coding region so that it can be used for the next step of translation. Finally, splicing happens. So what happens is there are non-coding regions in the DNA. Exons are coding or expressed areas. Introns are intervening sequences that interrupt that sequence. So basically, not like, as I said, there's some non-coding DNA in the middle of these coding regions. And this is again only in eukaryotes. So the purpose of splicing is to remove those non-coding regions from that messenger RNA. So basically what happens is this structure called a spliceosome, which is a structure made of both proteins and RNA, it sort of takes the messenger RNA molecule and it kind of twists the introns around those non-coding sequences and then just snips the introns out and splices the exons together. And so you can see on this picture, basically that intron is snipped, and what you're left is just the coding region. So again, it's kind of weird that DNA has these non-coding regions in them. We're going to talk more about that in our next chapter, but it does in eukaryotes, and so the spliceosome cuts those out. Some potential benefits, the answer is we don't really know. Well, actually, we do know at least some of the benefits of introns. One major benefit is it allows for alternative splicing of RNA. In other words, we can take these 5 exons and we can actually splice them in different ways, 1, 2, 3, 4, 5, to make one protein, or just 1, 2, 4, 5. Get out the 3 and make a slightly different protein. 1, 2, 3, 5 makes a slightly different protein. So by having these introns, it allows us to make multiple proteins from the same stretch of DNA. And this is much more significant than we ever thought. But time is out. So it also